In this second part, we will look at the basic function of the inner ear, and more specifically the cochlea, and what happens if this sense organ is damaged. The inner ear consists of both hearing and balance organs. The hearing organ is the cochlea, a snail shell-like, fluid-filled spiral tube. As the stapes moves in and out, the fluid in this tube moves backwards and forwards. The cochlear tube is divided down the middle by a partition, and the sense organ, the organ of Corti, sits on this membrane. This basilar membrane is narrow and stiff near the stapes at the base of the cochlea, and gets broader and floppier towards the tip of the snail shell, or the apex of the cochlea. This change in basilar membrane along the length is the basis for the cochlea being able to separate sound into different frequencies. If we were able to straighten out the cochlea, we would see that although the tube itself gets narrower towards the apex, the movable portion, the basilar membrane, gets broader and floppier. As the stapes moves rapidly in and out in response to sound, a travelling wave occurs along the basilar membrane. It is this up and down movement of the basilar membrane that stimulates the organ of Corti and causes the nerve fibre to fire. At the base of the cochlea, the basilar membrane is narrow and stiff and moves best to high frequency sounds. At the apex of the cochlea, the basilar membrane is wider and floppier and thus moves best to low frequency sounds. This means that the cochlea has a frequency gradation along its length, what is called a tonotopic organisation. If a single frequency sound is played into the ear, then it produces basilar membrane movement that is restricted to a particular place in the cochlea, and so only nerve fibres attached to that place will fire. If a different frequency sound is played, nerve fibres at a different place will respond. This is the basic mechanism by which the cochlea is able to separate sounds into different frequencies. In summary, because of the structure of the cochlear partition, the cochlea is tonotopic. Different locations along the spiral respond best to different frequencies. This is a property of the structure of the cochlea, and means that the organ of Corti, and hence the auditory nerve fibres nearest the stapes or base of the spiral, respond best to high frequency sounds, and regions near the tip of the spiral or the apex respond best to low frequency sounds. This means that the cochlea can separate sounds into different component frequencies. Within the cochlea and sitting on the basilar membrane is the sense organ of hearing, the organ of Corti. As fluid in the tube moves in response to sound, the basilar membrane and the organ of Corti move up and down. This movement stimulates the sensory cells within the organ of Corti, or the hair cells. The organ of Corti contains two types of sensory cells, or hair cells. One of these is the inner hair cells. These detect the movement and stimulate the auditory nerve to indicate that frequencies corresponding with this place in the cochlea are present in the sound. In order for these to... The second type of hair cells are the outer hair cells. These act like tiny amplifiers that increase the movement of the basilar membrane and the organ of Corti, so that it can be detected by the inner hair cells. Without these outer hair cells, the ear will be much less sensitive and some normal speech sounds will be inaudible. Two key aspects of the sound are the intensity of the sound and the frequency content. Sound intensity is a physical aspect of the sound that relates to what we describe as loudness. The higher the sound intensity, the more the basilar membrane moves and the more the auditory nerve fibres are active. For low intensity sounds, the outer hair cells are needed to amplify the basilar membrane movement enough for the inner hair cells to detect that movement. Without this outer hair cell amplification, the ear wouldn't be able to detect low intensity sounds such as whispers. Sounds such as speech are very complex, and as well as different intensities, they also contain many different frequency components. As we have seen, the ear is able to separate different frequencies from each other. The outer hair cells play a crucial role in this. By amplifying the movement of the basilar membrane, they make it easier to distinguish frequencies that are close together. They improve what we call frequency selectivity, our ability to separate out different frequency sounds.